diagram, myxin and also petromyxin. But cerebral hemisphere is small. Pineal body and parietal body is also well developed in petromyxin and absent in the myxin. Pineal apparatus, which is connected to the habenular ganglion, which is present in petromyxin and also myxin. Two optic lobes are present, but these are not divided or these are imperfectly differentiated. Medulla oblongata, which is well developed, and cerebellum, which is small, transverse, dorsal, band-like structure. Pituitary body is also well developed. Next, this is the diagram. We see cyclostomata and myxin, and this is the brain of cyclostomes. Then in the fish, comparison with the fish, in the, for example, shark or dogfish, olfactory lobes are enormously large. We see in the diagram first. So this is the brain of shark. So this is the part, upper part, it is called as a forebrain, then midbrain and hindbrain. So forebrain, it consisting olfactory lobe, then cerebrum, and then diencephalon. So olfactory lobes are sac-like, these are ends into the sac-like structure because of these are active swimming habit of fish. Then olfactory lobe, these are connected to the cerebral hemisphere by olfactory peduncle, lateral side of the cerebral hemisphere. This is the olfactory peduncle, one side, and this is the one side, which ends into the sac-like olfactory or fan-like structure. It is called as the olfactory sac, and these are the lobes. Olfactory lobe joins to the cerebral hemisphere, lateral side, by olfactory peduncle. It is also called olfactory tract. So olfactory lobe, last in the slide we see, it is called also renencephalon. Then this is the second part, is called as the cerebrum. In the fish, cerebrum is not divided, so it is called. In the other fishes, other animals, these are equally divided, so it is called as a cerebral hemisphere. But in fish, it is not divided, it is larger, and cavity of this cerebral hemisphere, it is called as a paracy or lateral ventricle. Then roof of the cerebral hemisphere, it is called as a pallium, upper surface, and lower ventral surface, it is called as the corpora striata. So second part of the forebrain, it is called cerebrum. Then third part, it is called as the diencephalon, which is not seen, diencephalon, because it is covered by cerebellum and also optic lobes. Diencephalon, anterior side, there is a presence of pineal body or pineal stalk, and roof of this diencephalon also consisting anterior choroid plexus. This secrete cerebrospinal fluid. Then ventral surface of this diencephalon, there is a presence of optic chiasma, cross of nerves, it is called as a optic chiasma. Below them, there is a presence of infundibulum, and outgrowth like structure, there are two inferior lobes, and below them there is a saxus vasculosus and hypophysis. So it is the ventral part of the diencephalon. Then midbrain. Midbrain, it consisting optic lobes. There are two optic lobes. Some part it is covered by cerebellum, and another part it is called as a also crura cerebra. It is not seen in this diagram. Then so optic lobes and crura cerebra, these are optic lobes are the midbrain part. Then third part, hindbrain. Hindbrain, in that cerebellum, it is also well developed, and it covers the optic lobe, and middle also there is a lower part, medulla oblongata, and well developed it is cerebellum, and also there in the lateral side there is a restiform body or corpora restiforma. The active swimming habit, which is also this assists this help to cerebellum. Then next part is the medulla oblongata. Medulla oblongata, it is broad anterior side and narrow at the posterior side. And this anterior roof of this, there is a presence of posterior choroid plexus. So there are two choroid plexus. One is the larger anterior choroid plexus, second one is the posterior choroid plexus, and these secrete cerebrospinal fluid. These are found in the ventricles. So this is the ventral part. This the diencephalous ventral part, infundibulum, optic chiasma, inferior lobe, then hypophysis, saccus vasculosus. These are the ventral part, and this is medulla. Then cavities of the brain. So this is the rhinocele. Olfactory lobe cavity, it is called as a rhinocele. Lateral cerebral hemisphere cavity called as a paracele or lateral ventricle. Then these two lateral ventricle unite together, and there is a formation of foramina monorum. Diencephalon cavity, it is called as a diocele. Then optic lobes cavity, it is called optocele. Then this canal, narrow canal, it is called iter. And cerebral hemisphere's cavity, cerebellum's cavity, epicele. It is not present other animals, but only in the fish. And this is the metacele or medulla oblongata's cavity, metacele. So this is the fish brain or dogfish brain. It is divided into forebrain, midbrain, hindbrain. Part of the forebrain, olfactory lobe, cerebral hemisphere, and diencephalon. Midbrain, optic lobes, and hindbrain, it consisting cerebellum and corpora restiform body and medulla oblongata. So these are the parts: dorsal side, dorsal view. This is the ventral view, and this is the ventricles or cavities of the brain. So rhinocele, it is the cavity of olfactory lobe. Lateral ventricle, these are a cavity of cerebral hemisphere. Then these two sides, lateral ventricle, unite together. It is called foramina monorum. Then diencephalon's cavity, it is called diocele. Optic lobe's cavity, optocele. Then narrow canal, it is called as the iter, or it is also called aqueduct of Sylvius. And cerebellum's cavity, only found in the fish, it is called as the epicele. This is the epicele. And medulla oblongata's cavity, it is called as a metacele. So this is the, then we see in the theory, in elasmo branch or shark or dogfish, olfactory organs are enormous, large, attached to the cerebrum by short olfactory tract or peduncles. We see in the diagram. Optic lobes are moderate in size. Then sensory organs, these are also saccus vasculosus, which is present into the ventral surface of the brain. Pineal apparatus, it is well developed. Cerebellum is large due to the active swimming habit. And also there is a presence of corpora restiform bodies. So this is the brain of fish. Then in the another ostick thighs, elasmo branch, it is the order. Next order is the ostick thighs or bony fish. So in bony fish, brain is specialized than the elasmo branch or dogfish. In perch, there is an olfactory lobe and cerebral hemisphere. Diencephalons are similar. Perch is a fish, and in perch, olfactory lobe, cerebral hemisphere, and diencephalons are similar. Optic lobes, cerebellum are larger than in shark or dogfish. In some bony fishes have restiform bodies, and in bottom feeder having anterolateral, there is a bulging or vagal lobes. These are found in the, this is the diagram, this is the vagal lobes. These are found in the, this is the olfactory lobe, cerebral hemisphere, optic lobes, cerebellum, and these are found only in the vagal lobes, only in the bottom feeding fishes. So these are the found in the fish. Then we see the amphibia. Amphibia, well-known example is the frog. So in frog, we see in the diagram first. So this is the brain of frog. In brain of frog, it is also divided into the forebrain, midbrain, and hindbrain. So forebrain, it consisting olfactory lobe. Then this is the cerebral, equally divided part. It is called as a cerebral hemisphere. In fish, we see only cerebrum because these are not divided. But in frog, it is divided into the equal part. Anterior, narrow, posterior, it is broad. Then third part it is called as the diencephalon. It is also covered by anteriorly cerebral hemisphere, posteriorly, and it is by optic lobes. So diencephalon, there is also pineal body is present. Then anterior, the roof of this diencephalon, it is called anterior choroid plexus, and ventral surface of the brain is showing optic chiasma. Then also infundibulum and pituitary, these together called as a hypophysis. So these are ventral part of the frog brain or amphibian brain. In that also there is a cavities. So this is the olfactory lobes cavity, it is called rhinocele. Then lateral side cerebral hemisphere consisting cavity, it is called lateral ventricle. These two unite together, it is called as a foramen of Monro. And this in the diencephalon cavity, it is also called third ventricle, and optic lobes cavity, it is called optocele, and narrow canal, it is called ether or aqueduct of Sylvius. We joins the medulla oblongata's cavity, it is called metacele. So this is the optic lobe cavity, optocele. Medulla, midbrain parts are optic lobes, and also there is a presence of crura cerebra, nervous tissue band. These are present. These are called crura cerebra, and hind brain consisting cerebellum. In frog brain, cerebellum is not well developed. In fish brain, we see cerebellum is well developed, but in frog, it is not, because frog is a not active animal, and cerebellum is transverse band-like structure, and it is not well developed. Then roof, this is the medulla oblongata. Medulla oblongata broad at the anterior side and narrow at the posterior side. A roof of this also medulla oblongata, it is called as a posterior choroid plexus. There are two choroid plexus. One is the roof of diencephalon, another one is the roof of medulla oblongata. And these are anterior diencephalon's roof, it is called as a anterior choroid plexus, and in the medulla oblongata it is called posterior choroid plexus. These two choroid plexus secrete cerebrospinal fluid. These are found in the ventricles. So then these are the cavities of the brain. Cavity of olfactory lobe, it is called rhinocele. Then these are the lateral ventricle first and second, it is also called paracele. These two lateral ventricles unite together called foramen of Monro. Then it joins to the diencephalon cavity, diocele or third ventricle. Then cavity of the optic lobe, it is called optocele. This cavity joins to the medulla oblongata, it is called as a narrow canal-like structure, ether. And medulla oblongata's cavity, it is called metacele or fourth ventricle. So this is the amphibian brain. So we see in the brain of frog shows small olfactory lobes, large optic lobes. Optic lobes are probably dominate the cooperating center in the amphibian brain. Two cerebral hemispheres shows greater development in accordance with more complex activities of locomotion, hibernation, and breeding. So poor development of cerebellum, transverse band-like structure, it decrease in muscular activity because frog is not active anymore. Then medulla is also small, and small pineal body is present in all the modern amphibians. So we complete this amphibian brain or frog brain. Next one is the reptilians. In the reptilians, for example, lizard. In lizard, we see first in the diagram. So this is the brain of a lizard. So this showing upper part, forebrain, it consisting olfactory lobe. Then this is the cerebral hemisphere and diencephalon. Then midbrain, there is a presence of optic lobes, and hindbrain consisting cerebellum and medulla oblongata. So in this, there is a different from the amphibian brain. So olfactory lobe, these are two bulb-like structure, anterior side. Then olfactory lobe joins to the cerebral hemisphere by a thread-like structure. It is called as a olfactory peduncle. It is not present in the frog brain. So then in the cerebral, it is anteriorly narrow, posteriorly broad, which covers the diencephalon. Then the this is the diencephalon compressed area which is covered by optic lobes and cerebral hemisphere. Then the diencephalon, in front there is a pineal body and anterior choroid plexus. Then ventral surface of the diencephalon, there is presence of cross of nerves. It is called as a optic chiasma, hypophysis, infundibulum and hypophysis, together formation of pituitary body. And then midbrain, it consisting optic lobes, two optic lobes. It is also called corpora bigemina because of number 2. Then crura cerebra, a longitudinal band. It is seen in this diagram. Longitudinal band, nervous tissue band. It is called as a crura cerebra, which conduct impulses from hindbrain to the forebrain. Then third part, hindbrain. In the hindbrain, this is the medulla oblongata and cerebellum. Cerebellum, it is also not well developed in the reptiles. Then this is the cavities. Olfactory lobe cavity, it is called rhinocele. Cerebral hemisphere cavity, paracele or lateral ventricle. These together unite, it is called as a foramen of Monro. Then diencephalon cavity, it is called diocele. Optic lobes cavity, optocele. Narrow canal is called iter, and it joins to the medulla oblongata's cavity. It is called metacele. So these are the cavities of the brain. So the reptilian brains show advancement than the amphibians because these animals are completely terrestrial mode of life. Then telencephalon increases. Telencephalon means cerebral hemisphere increase to become the largest regions of the brain. Then two olfactory lobes are connected to the cerebral hemisphere by olfactory peduncle. Then pineal body, in reptiles only there are parietal body and pineal bodies are present. Only in Spinoda and these are absent other reptiles. So parietal organ, it is also called third eye, and it is present only in some animals or reptiles. Then third ventricle also reduced. Cerebellum is pear shaped and larger than amphibians. Then we see this structure in also fish, in amphibia and also reptiles. Now all these are parts are present, but functions, what is the functions of that brain part? So olfactory lobe, it is the sense of smell. Then cerebral hemisphere, it is the controls voluntary activities. For example, willpower, then memory, intelligence. All activities are controlled by cerebral hemisphere. Then diencephalon, it recognize the actions like heat, cold and pains, or these are controlled spontaneous actions of body. Then midbrain, it is optic lobes. Optic lobes function sense of sight. And crura cerebra, second part of this midbrain, it is the longitudinal nerve tissue band, and this nervous tissue band, it is conduct impulses from hindbrain to forebrain. Then third part or hindbrain, it consisting the cerebellum and medulla oblongata. Cerebellum, it is the body, maintain body equilibrium, and medulla oblongata, it controls involuntary activities like respiration, heartbeat control, then also excretion. All these body activities, these are which are not our control. These are controlled by the medulla oblongata. So involuntary activities, these are controlled by the medulla oblongata. I can repeat this again. Olfactory lobe, function of olfactory lobe, these are sense of smell. Cerebral hemisphere control voluntary activities, for example, intelligence, willpower, these are controlled by memory, these are controlled by cerebral hemisphere. Diencephalon, it also controls spontaneous actions of our body. Optic lobe controls sense of sight. Crura cerebra, it controls or conduct impulses from hindbrain to the forebrain. Cerebellum, it is maintains body equilibrium. And last medulla oblongata, it controls involuntary activities like respiration, excretion, heartbeat control. These are all are controlled by the medulla oblongata. So these are the, we complete only the first one, fish or cephalochordate, for example, amphioxus, then cyclostomes, then fish, then amphibia and reptiles. So these are the comparison in cyclostomata. In cephalochord, first protochord is cephalochordate. For example, amphioxus. Brain is not well developed, only anterior part of the brain is present, and it is called prosencephalon or cerebral vesicle with also single ventricle. Then in cyclostomata, for example, in the tetromyzon and myxin, brains are not divided into subdivision, perfectly subdivisions. These are divided imperfectly. For example, it is consisting olfactory lobe, cerebrum, it is smaller. Then this pineal body joins to the habenular ganglion, and diencephalon is also present. All parts are present but are not divided equally or imperfect. These are divided imperfectly. Similar in this also cyclostomes. Some parts are well developed, some parts are larger, some parts are smaller. So in the cyclostome, brain is primitive, subdivisions are not well marked, olfactory lobes are prominent, but cerebral hemispheres are small. Pineal body, parietal body are well developed in petromyzon, absent in myxin. Then pineal body connected to the ganglion, these are called habenuli ganglia. These are present only in the cyclostomata, not other animals. Optic lobes are present, but these are imperfectly divided. Medulla oblongata also well developed. Cerebellum is smaller and transverse band-like structure. Pituitary band is also well developed. Then in fish, we see in the fish shark or dogfish. So these are the part of the brain. First one, olfactory lobe, it is called renencephalon. Second, cerebrum, it is called telencephalon. Diencephalon called thalamencephalon. These are included into the forebrain or prosencephalon. Then midbrain part consisting optic lobes. This included into the midbrain. It is also called mesencephalon. And hindbrain, it consisting cerebellum. It is called metencephalon and myelencephalon, and the part of the rhombencephalon. Then brain consisting olfactory sac, these are well developed in the fish brain, and also cerebellum is also well developed because of its active swimming habitat. In fish brain, these are similar, some parts are also well developed. Then in amphibia, olfactory lobes are not well developed, but because of this, in fish it is developed, but in frog, olfactory lobes are not well developed. Then, but cerebral hemispheres are well developed, and these are equally divided into two equal halves. So it is called as a cerebral hemisphere. But in fish, we say in this there is a cerebrum, and in frog it is called cerebral hemisphere. Then it is also similar to this prosencephalon, mesencephalon, metencephalon. Olfactory lobe, these are anterior side joins to the cerebral hemisphere. Cerebral hemisphere, these are larger. Roof of the cerebral hemisphere, it is called pallium, and ventral part of it is called as a corpora striata. Then third part of the prosencephalon, it is called diencephalon. Diencephalon, the roof of this diencephalon, anterior choroid plexus, which secrete cerebrospinal fluid, and in front there is a presence of pineal body. Then diencephalon, it is compressed part which is covered by cerebral hemisphere and also optic lobes. Then midbrain or mesencephalon, equally divided optic lobes are present, and these functions of the sense of sight. And there is another part, longitudinal nervous tissue band. It is called as a crura cerebra, which is seen in the ventral part of the brain. Then diencephalon ventral part showing cross of nerves. It is called as a optic chiasma. Below them there is presence of infundibulum and pituitary or hypophysis. And cavities of the brain, it are similar in the fish, also in the frog, and also in the reptiles. So this is the reptile, which is the difference in that there is a olfactory peduncle is present and cerebral hemisphere, because these animals are terrestrial animals. So this cerebral hemisphere also well developed, and olfactory peduncle, this bulb, olfactory bulb joined by cerebral hemisphere. These are the two thread-like part. It is called as a olfactory peduncle, which is present in the reptiles. Then parietal body and pineal bodies are present. In parietal body, it is called as a third eye, which is present in the some spinodon only, and some modern fishes these are absent. Pineal body and parietal body, these are endocrine in function. So this is the forebrain, olfactory lobe, cerebral hemisphere, and diencephalon. It is covered by the cerebral hemisphere. Then diencephalon, it is compressed in front. There is a presence of anterior choroid plexus, and this cover, it is secrete the cerebrospinal fluid. The diencephalon ventral surface showing the cross of nerve, optic chiasma, then infundibulum and pituitary or hypophysis. Midbrain, these are the optic lobes, these two optic lobes and sense of sight. From this optic nerve, anterior side cross ventral surface, and there is formation of optic chiasma. Then hindbrain, there is a diamond shaped structure. It is called as a cerebellum. It is the poorly developed, not well developed. And second part, medulla oblongata. Medulla oblongata, it is broader anterior side and narrow at the posterior side, which joins to the spinal cord. So this medulla oblongata, it is also anterior part showing the posterior choroid plexus, and this choroid plexus, it secrete the cerebrospinal fluid. So these are the cavities in this olfactory lobe or olfactory bulb, cavity called rhinocele. Then cerebral hemisphere cavity, paracele or lateral ventricle. These together unite formation of foramen of molar. And then diencephalon cavity, diocele. Optic lobes cavity, optocele. Narrow canal is called ether, and this joins to the metacele, medulla oblongata's cavity. So this is the cavity consisting cerebrospinal fluid. Cerebrospinal fluid, posterior choroid plexus nourishes brain, nourishes part of the posterior part of the brain, and anterior choroid plexus nourishes anterior part of the brain, and it prevent or make it is from the protect from the mechanical jerks. So brain of, so we complete this half part of the development of brain comparative account. We see in the cephalochordate, cyclostome, then also fish, then reptiles, amphibia and reptiles. Remaining part we see in the next unit. Hello?
Yes, ma'am.
हाँ, ऐकायला येते? कळलं का सर्वांना? काय शंका आहे का तुमची? समजलं सर्वांना? हॅलो, अनम्यूट कर सगळ्यांना ते.
येस मॅम.
हा, सगळ्यांना कळलं का व्यवस्थित?
हो मॅम.
व्यवस्थित समजते ना? कारण हा जरा पाठ अवघड आहे. कुणाला काय शंका असेल तर मला विचारा. हॅलो, सांगा एकीने बोला पाहिजे तर.
शॉर्ट नोट
अगं हे कंपॅरेटिव्ह अकाउंट कंपेअर विथ आता फिश ब्रेन विथ फ्रॉग ब्रेन और फिश ब्रेन विथ द रेप्टाइल ब्रेन और रेप्टाइल ब्रेन और द फिश ब्रेन असं हे कंपेअर करायला तुम्हाला फुल क्वेश्चन विचारला जातो याच्यावर. त्यामुळे डायग्राम पहिल्यांदा आणि नंतर हे पॉइंट्स फोर ब्रेन मिड ब्रेन हाइंड ब्रेन त्याचे पार्ट त्याचे फंक्शन मग कुठला डेव्हलप आहे पुन्हामध्ये कुठला डेव्हलप आता मी सांगितलं का नाही. आता फिश असेल तर फिश मध्ये ऑल्फॅक्टरी सॅक इट इज वेल डेव्हलप. फ्रॉग मध्ये वेल डेव्हलप्ड आहे का नाही. फिशला का गरज आहे फिश इज दी ऍक्टिव्ह स्विमर होय की नाही त्यामुळे ऑल्फॅक्टरी सॅक हा चांगला डेव्हलप असतो. नंतर फ्रॉग मध्ये एवढी त्याला गरजच नाही फ्रॉगला तो पार्ट तिथे वेल डेव्हलप्ड नसतो. आणि नंतर रेप्टाईल मध्ये पण तो पार्ट आहे काय वेल डेव्हलप नाही. म्हणजे असं कम्पेअर करायचं आहे आपल्याला एक पार्ट आणि त्याचं. आता फिश असेल फिश ब्रेन इज कवर्ड बाय कार्टिलेजिनस मेम्ब्रेन. फ्रॉग ब्रेन असेल तर फ्रॉग ब्रेन इट इज कवर्ड बाय बोनी कार्टिलेज और बोनी इट इज कॉल्ड क्रॅनियम. असं एक कम्पेअर करत जायचं आहे 1 to 1 to 3 असे पॉइंट्स तयार करायचे आणि त्यानुसार कम्पेअर करायचे. आले लक्षात सगळ्यांना कळलं का मला सगळ्यांनी सांगा.
हो
हो सगळ्यांना समजलं ना. प्रेझेंटी हे केलीस का प्रेझेंटी तयार केली.
हो मॅम हे रेकॉर्ड झालंय ना लेक्चर ह्याच्यामध्ये नावं दिसतात.
दिसतायत का पण एक स्क्रीनशॉट काढून मला पाठव मला ते आता द्यायचंय पाठवून.
अर्पिता स्क्रीनशॉट काढ ना.
किती जण होते आता.
आता 10 जण आहेत मॅम लेक्चरला.
14 जण होते ना मगाशी. नाही किती जण 10 जण आहेत का.